All right. Oh, thank you. It's a great pleasure and honor to be here today and to receive the prize. It's the first time that somebody from the University of Gothenburg from the field of chemistry has been given this award, so that's great. I've been working for a little more than 15 years in the field of membrane protein structure and dynamics. So we work on the field of membrane proteins. Membrane proteins are proteins that perform functional roles inside a cell membrane. So here we have a picture of a eukaryotic cell, and you can see there's these membranes surrounding. There's the outer membrane, the plasma membrane, which surrounds the cell. There's different membranes inside surrounding the nucleus and the mitochondria. And these membranes are formed by lipid bilayers, and inside the lipid bilayers there's lots of proteins which perform all sorts of functional tasks. Cell signaling, sensory perception, communication, energy transduction, and they're essential for cells to live. Now, one of the membrane protein families that I've been working on for quite some time is an area of photosynthesis. So we all understand photosynthesis. It's the basic concept of how the energy of sunlight is captured by cells, captured by proteins inside a cell, and then converted into a chemical energy that can be used by the cell. So a conserved mechanism of photosynthesis is responsible for about 90% of the energy input into the entire biosphere. So all life on Earth has to be thankful for basic energy chemical reactions that are governed by membrane proteins governing photosynthesis. Now, my lab isn't working on proteins from sunflowers. A little less spectacular than that. We're working on purple bacteria, proteins isolated from purple bacteria. So these purple bacteria grow in oxygen-deficient environments in lakes. And they look purple because they're full of membrane proteins that harvest light, and those membrane proteins are colored purple. In our lab, we culture these proteins. We can then purify them, take them out of the membrane. We can make crystals of them or study them in the solution phase. Now, we use X-ray methods, so we travel to extremely brilliant X-ray sources. When I first began learning about X-ray crystallography, I traveled to the European Synchrotron Radiation Facility, which was a world-leading stage for X-ray science for more than 20 years. Sweden is investing in Max IV Laboratory in Lund. It's a synchrotron currently under construction. It's going to be the most brilliant X-ray source in the world when it's complete. It's an exciting project. It's very much going to have scientists from all over Sweden involved in Scandinavia. And of course, structural biology is going to benefit. I've also had the pleasure and the privilege to be one of the first users of X-ray free-electron laser radiation in life science. And the world's first X-ray free-electron laser was in Stanford in the USA in California. And rather than the electrons moving around in a circle with synchrotron radiation, the free-electron laser delivers electrons along a linear accelerator here, and it produces extremely intense, very, very short bursts of X-rays. So the step from what you can achieve in terms of a number called peak brilliance, which is how many photons you can focus onto a small spot in a short time, that step is 9 orders of magnitude greater from a synchrotron source to a free-electron laser source for a very short time. And that creates completely new opportunities for studying biomolecules and other areas of science, including physics and chemistry. So this shows an experiment that we've done using an X-ray free-electron laser. We produced a sample produced at home in Gothenburg, and here's samples of photosynthetic reaction centers, photosynthetic proteins from a membrane from bacteria isolated in the solution. We then deliver these across the X-ray beam, in this case using a tiny jet which is about 10 micrometer across. We have X-ray pulses from the free-electron laser and a laser pulse to excite the protein, to start a reaction inside the protein to simulate the reaction that we have caused by sunlight. And then we scatter the X-rays from our sample onto an X-ray detector, record these, and try to see how that X-ray scattering changes with time so we can understand how the protein is changing in time. And one of the results that has come from my lab, which is exciting. Is this going to work? Okay, I have to click. Oh, I've lost it. Maybe if I go like this. No? Okay, it didn't work. There was a movie. Why didn't the movie work before? That's a pity. one of the results is that we get a breathing motion in the membrane protein on a time scale of picoseconds. It breathes out and in. And that motion happens on a time scale, a very fast time scale, the time scale that takes the speed of light to travel just a millimeter. We get this motion inside the protein we could observe. And then that motion was completely damped on a time scale that it takes light to move just a few centimeters. So there was an ultra-fast breathing motion. And our challenge is to understand if this ultra-fast breathing motion is important to the fundamental reactions of photosynthesis. So there's ongoing work in our lab to try to explore that in more detail. Okay, I like to think about that motion as being analogous to when a baseball strikes a mitt in a game of baseball. And then you have the impact, you have energy focused at a certain point, and this energy is diffused throughout that structure of the glove of the mitt to protect, and that energy being diffused protects the hand from being damaged. So there's a kind of a mechanism here where energy is entering into light harvesting proteins and being diffused through structural motions, a protein quake on a very fast time scale. And the extent to which this can be coupled to protein function is an ongoing question. I can't, in 5 minutes, I can't thank all of the people who have involved in the work that we've done for the last 20 years, who have been very much involved in international collaboration. A lot of people have come through my lab. But of course, there's been very important support from external financing, particularly the Research Council. You were actually in charge of SFF when I got my first SSF grant. And recently, tremendous support from the Wallenberg Stiftelse in particular. So thank you for your attention.
Thank you very much.